We are now recording. Even though we won't be starting until about 4 or 5 minutes from now. Since we are recording, anybody has questions, feel free to ask. That way, anybody who's not here, if they miss the questions, they can always watch the video. Good morning. All right, less than 2 minutes. Start attendance. Brumfeld, not here yet. Champion is here. Gorby is not here. Actually, don't worry about that. Gross is not here. Gunther is here. R Hill is here. Z Hill is not here. Jackson is not here. Keys is not here. Perez is here. Sigmund is here. Slaughter is not here. Smith is not here yet. Stover is here. Tompkins is not here yet. Neither is Vanest. Watson is not here yet. Young is here. Zweiback is here. Lawrence is here. Wait, no, Lawrence is not here yet. Wilson's not here. I'll go through it one more time here shortly. Excuse me. All right, 8 o'clock. One more time. Well, no, that's not good. Let's not do it one more time. Keys is here, right? Yep. So just to be clear, for the recording, one more time. Champion is here. Gunther is here. Randall Hill is here. Was here first. So we'll actually go for that. Keys is here. Perez is here. Sigmund is here. Stover is here. Young is here, Ryback is here, and that's it, right? Did I miss anybody? All right. It's almost 8:01. Shut the doors when the time comes. So I'll do the normal announcements in the meantime, the stuff that's completely repetitive. So I apologize for you guys probably getting sick of hearing it by now. But at the end of the semester, when someone says, I didn't know I have to do this, that way I can say, well, I said it every time we met. So attendance is mandatory. Participation is great. Actually, that is new now that I think about it. I'm not necessarily rewording it, I'm just emphasizing the wording. So if you remember back when we met on the first day of class, I said I have to turn in attendance, and that's whether you're here or not. That's all the university cares about. Then I said there's actually participation points, which you earn. And I said, generally speaking, if you're here, you automatically earn it. But you have to do something to earn it. So even if you're absent, excused or not, and I haven't been enforcing this, so I don't feel like I emphasize this enough on day one. Even if you're absent, you still need to get a participation grade for the day. So if you're absent, excused or not, you'll need to watch the video. If there's not a video, then we'll find something else for you to do to get your point for the day. Let me know. We'll cross that bridge when we get there. Hopefully we don't need to. But anyway, today's first word for attendance is CC. It's 2 letters. CC is in chubby checker.
So we miss because of something, we still have to watch the videos?
Yeah. Because that's your participation points. Or, like for some of you have so many extra credit points because at the end of the end of every lecture, you guys are sending in the words, and it seems like it's a little bit, but some of you have got so many that you don't even you have more points than you need, and it makes up for the points for when you're gone. So it's up to you. So when you're gone, we can look at your points, individually, and I can help tailor a plan for you because you might not even need to worry about it. Besides, you should be watching the lectures anyway. It's going to help, right? Anyway, so yeah, CC is the first word for attendance. That's in chubby checker because that's his birthday. So any questions so far? All right, to remind you, the meiosis pre-lab is due today. This is a heads up, so you're not surprised about it. There's 2 sets of video questions. Speaking of which, when you answer the video questions, you're in section 101.03, it'll ask you that. That'll be very helpful. I have 218 students this semester, and you guys aren't the only class I'm teaching, and I'm not the only professor using those forms, so it's very helpful for you to use that. There's an intro, and then questions about the introduction. Keep in mind that those questions are basically evidence that you read the introduction, so don't look up the answers anywhere else. Just make sure the answers come from the introduction. And a little hint, this is the most commonly missed question, and we haven't got to it in lecture yet, so I can see why it's missed. Well, you know what? I'll give you that hint once we get there, because we're going to get there in lecture today. So anyway, yeah, 2 sets of video questions. Introduction questions. There's no hypotheses this time. Again, like I mentioned on Wednesday, this is an interesting lab in that we're not doing a true experiment where it's like you're testing a hypothesis. This is more of like an exercise that help you get more familiar with mitosis and meiosis. It's 100% virtual, so you need to bring a computer next week. That also means you can get a head start if you want. So while you're doing the pre-lab, if you feel like I'm going to keep going, you can do the whole lab if you want, or you can just do part of it if you want. If you want to get an early start so you don't have to spend so much time in lab next week, that is completely okay. And even though you're getting an early start, and yes, I'll be here to teach you on Wednesday how to do it, I can still help you. Like if let's say you want to do it today at 11 a.m., which is during my office hours, you can say, hey, can you help me just get this lab done? Meet me online, I'll help you do it. Any questions about that? All right, do not copy. I'm reiterating this. I'm going to keep on saying it, and it seems to be with the same group of people. Don't copy. Do not copy. Or if you are going to copy, at least use your brain. So there was one really ridiculous pre-lab question from last week. It was a hypothesis about light intensity. And someone said, yes, I think light intensity affects photosynthesis because of all the work they have to do. And I'm like, that sentence makes no sense. And then other people all had that same exact wording because of the work they have to do. So again, don't copy, but if you're going to, at least think about what you're writing. Read the question, read what you copied, and if it doesn't make sense, say, ooh, maybe I should think for myself, or maybe I should tell the person that I copied from, hey, that's a ridiculous answer, maybe think about rewriting it. So do not copy. Let me jump ahead to this bullet point. I'm here to help you. I want to help you. I prefer to sit with you online and walk you through each question, because I don't just want to make sure you get a grade or, I want to help you. I want to make sure you understand it. That's my job, is to teach you, not to test you. it were up to me, I could just do this on the honor system and we wouldn't even need pre-labs or lab questions. I'm like, come on, let's just all learn together. But unfortunately, obviously, because people are copying, most of them, some people are just here for the grade. I was too, sometimes, as an undergrad, so I'm not saying that to judge them away. I've been there. Anyway, please don't copy. Please let me help you. That's what I'm here for. Any questions about pre-lab? All right, again, just to remind you, technically, labs are always due on Wednesday before you leave, but they are past due on Friday. So make sure you get lab sick, the previous lab in today. Again, let me help you if you need help. These aren't questions, these aren't quizzes or exams, right? So you're working on it today, but it's, haven't looked at it since Wednesday, you need me to refresh your memory, like, what does this data mean? Let me know. Also, a common mistake, sorry, I know I'm going on and on today. Common mistake I see with this that I mentioned on Wednesday, when you calculate the total change of something, don't overthink it. Don't think about it as like it's a math class. Just look at common sense. If you go from like 7 to 9, that's a +2 change, right? If it went up. If you go to 10 to 9, that's a -1 change, right? So don't overthink things. Look at the big picture, and let me help you. Any questions about the lab that's due today? Okay. Exam 1, again, you can retake it as many times as you want. Open book, not timed. You can even help each other. I wouldn't say copy, but you can help each other. And then once you're done, make sure you email me, let me know. Otherwise, I won't change your grade because I won't know what needs to be changed. So retake the exam, let me know. You have nothing to lose. Your grade will definitely not go down. You can do that until exam 2 is closed, which is on October 12th. So again, exam 2 is also open. It covers everything prior to the chapter that we're doing today. It covers chapters 3, 4, and 5, right? Cells, respiration, photosynthesis, energy, basically. So any questions about announcements, reminders? Does anybody have their own? Okay, please let me help you. That's what I'm here for. All right, let's jump back into it. We're done talking about energy. Well, we're done for the big discussion about energy, right? That's the previous exam. We are now on to new stuff. And this new stuff, this new exam that we're going to take, exam 3, when we eventually get there, that's mostly going to be about reproduction. So what we're talking about right now is the cell cycle, right? So first, before we even got to this, we talked about like some background information, right? So now you're a little bit more familiar with chromosomes and what they're made up of. And then I told you the cell cycle is basically the life cycle of the cell, starting from when it's first, I don't want to use the word born because it's not born, when it's first created from a previous cell, that's the beginning of the cell cycle. And the end of the cell cycle is when it itself splits into 2. So that we've already covered. What we left off was me breaking this down and saying the cell cycle is broken down into 2 parts, and you need to know these 2 parts: interphase and mitotic phase. And you can see here, obviously, there's a lot more than 2 parts. But my suggestion, and you can do it however you want, but I suggest learning the big picture and then start adding the details. So once you memorize the fact that there's an interphase and the mitotic phase, then you can start looking at these other parts. But for now, let's just leave it at that. Interphase and mitotic phase are the 2 parts of the cell cycle. Any questions about that? All right. And the way I remembered it as a student when I was a freshman is that, in this chapter, even though, how do I put this? We're talking about the whole thing, but mostly we're focused on the mitotic phase. Like for the exam, it's mostly going to be about the mitotic phase, specifically mitosis, which is part of the mitotic phase, and we'll talk about that later. So the way I remembered this was that, the mitotic phase is what it's all about, the exam anyway. And I remember that the interphase was that stuff that was in between. I don't know, that kind of helped me remember. But anyway, we'll move forward. There's no questions about this. Okay. So, as you can see in that last picture, and in this picture, and in the writing on the wall, so to speak, the cell is mostly in interphase. So that might be a test question too. Of the 2 phases, which one is the cell usually in? It's usually in interphase. Now, granted, the book does say 90%. I'm not going to ask you that number. I might use that number in a question. Like I might say something like, the cell is usually, 90% of the time, the cell is usually in this phase, right? And which would be the interphase. But I won't have a question like, the cell is usually an interface. What percentage of the time will it be an interface, right? So you don't need to memorize that number, just know that it might come up as part of the question and know for sure that the cell is mostly in interface. So what's happening during interface? We might get some questions about that. And again, like I said on Wednesday, towards the end of this chapter, you're going to learn a little bit about cancer. And to truly understand how cancer works, you need to understand how the entire cell cycle works. So this is really important stuff, not just for your exam, but just for your everyday life. Because unfortunately, one day you might have to do some research on cancer because you or somebody you love might have it. And all cancers are a little bit different, so it's good to have this foundation of knowledge that you can build from without starting from scratch. So anyway, during interface, cells perform normal functions, right? Basically, they're just doing the regular everyday cell things. You know, different cells have different functions, whether it be a kidney cell or a liver cell or a cell in your eyeball or a cell in your brain, right? They all have different functions. Whatever the function is, when they're in interface, that's usually what they're doing. They're just doing whatever their function is. But as we know from previous discussions, the whole idea sort of the cell cycle is to eventually split into two new cells. So the cell knows, or not knows, it doesn't have the conscious, but the mechanisms in the cell know that eventually it is going to need to be split into two. So because of that, as it's doing its normal function, it is also slowly getting ready to do that, right? So it's going to double everything inside of its cytoplasm. Because if you take one cell and cut it in half, then you would have two half cells, basically, right? But you don't want that. You want to have viable cells. So the cell that's eventually going to split needs to double its size. Or not double, but it gets larger. It's starting to double everything inside, right? All those organelles that you learned about in chapter 3, right? You got to double all those, right? So that way your new cells have what they need. Any questions about this slide? Okay, next. Let's talk about the different parts of interphase. So again, the whole cell cycle is broken down into interphase and the mitotic phase. So right now we are focused on interphase. I just told you on the last slide what basically happens during the entirety of interphase. Now we're going to look at the 3 different parts of interphase and what happens in them. For the most part, I'm not going to ask you any questions about what happens in the particular phases. You definitely need to know the order. So before I even talk about what happens in each one, I'll tell you, when we're talking about interphase, it's broken down into G1 and then S and then G2. So you definitely need to know that. Definitely need to know the order, the 3 parts of interphase and the order that they go. And I'll maybe ask you some questions about what happens during these 3 stages. And hopefully this will be common sense to you. Not common sense necessarily. Hopefully it'll be intuitive. So as I'm describing all this, hopefully you'll think to yourself, oh yeah, that would make sense for this to be the first part. And that would make sense for it to be the second part. So let's look at it. Obviously, it's part of interphase. That's the discussion we're having right now. Obviously, since S phase is next, it is before S phase, so that part is kind of intuitive. At this point, each chromosome is single, which reminds me, we had a conversation, actually might as well just show you, towards the end of the last lecture, we talked about this, right? When a chromosome does duplicate, which eventually it's going to need to before it can split, you have this single chromosome, and then it doubles itself, and those two, now it's considered 2 sister chromatids. They're identical to each other. They're still attached, but eventually they get separated. So what I'm saying is that in G1, there's still a single chromosome. So this is the situation for G1. Just one single chromosome that has not duplicated yet. Okay, there we go. And again, at this point, as in most of interphase, it's just doing whatever its normal function is. Whatever kind of cell it is, it's just doing that. It is growing a little bit, right? It is accumulating stuff, right? So it's accumulating energy, it's accumulating materials. But for the most part, G1 is just like completely just business as usual. It's not necessarily preparing much to do anything yet. Business as usual is what you think of for G1, which makes sense, right? Because again, I'm hoping this is intuitive. This is when the cell was first created, right? It's the very beginning of its life cycle. So it's as far away as it can be from splitting into 2 new cells. So it's, it's preparing a little bit for splitting, like this last bullet point shows, but for the most part, it's not doing much to prepare for that. So any questions about G1? All right. The big one, as far as we're concerned, for this discussion, for this chapter, when we're talking about mitosis and meiosis, right? Again, right now we're talking about the entire cell cycle, but as far as the exam is concerned, this is our main focus, is the mitotic phase. So as far as that discussion is concerned, this next part is really important. S phase is very important. Now, granted, as far as life is concerned, all the phases are equally important. But if we're having a discussion about mitosis or meiosis, then S phase is the most important one, in a sense, as far as the exam is concerned. Again, this is still part of interphase, so I'm just reminding you of that in writing. Again, regarding cellular reproduction, this is the most important part of interphase. Again, that's not going to be a test question. Which one's the most important? I'm just saying, once you get to the exam and we're asking questions about the different parts, you can imagine S phase is going to have more questions about it than G1 or G2. For this conversation, S phase is more important. At this point, the centrosomes are duplicated. And I only included that. I got a little star. Let me remind myself. Yeah, later you're going to learn about mitosis. You're going to learn about something called a mitotic spindle. The mitotic spindle, as you're going to learn later, is the thing that's moving everything around. Those mitotic spindles come from these centrosomes. So it's an important part of the story, and that's why I put it up there. That's in your textbook, so I put it up there. But as far as the exam is concerned, I'm not going to ask you when the centrosome is duplicated. What is important is this bullet point right here. This is when the chromosome duplication occurs. So again, that picture I showed you that had that single chromosome, that was from the previous phase. That was from G1. It is now in S phase when we duplicate that and we get our two sister chromatids, right? That's when the DNA copies itself. And then in a different chapter later, maybe the next chapter. Anyway, in one of the upcoming chapters, there's going to be a whole discussion about that. Right? You're really going to learn exactly how the DNA does that. But for now, you don't. For now, just know that it does that. It duplicates itself. That's when we get the sister chromatids. And the way that you can remember that is S, and S phase stands for synthesis, as in DNA synthesis. Because again, as far as we're concerned, that's the most important part other than mitosis. And S phase stands for synthesis. This is when we are synthesizing the new DNA molecules, which is going to be important. Because again, once we start talking about mitosis, there's a lot of stuff happening, but even then, we're mostly just considered focusing on what happens with the chromosomes. Mostly. We'll have some other side conversations, but for the most part, we're always talking about the chromosomes. Same here. S phase, the most important bullet point on here is that one. We're duplicating the chromosomes. All right. The next word for attendance, I'll do some more initials. LB. And Lindsey Buckingham. It is his birthday today, too. That's two musician birthdays. So any questions about S phase? All right, let's go on to the last part of interphase, which is G2. That's nice and easy to remember, right? G1, S, G2. Hopefully that's easy to remember. Again, it's part of interphase. It's in writing to remind you. Obviously, since we just talked about it, it's after S phase. Obviously, and again, I'm hoping this is intuitive for you, since I just said in S phase that the chromosomes have duplicated and we created sister chromatids, and hopefully, obviously, you would understand that in G2 phase, they're still like that, right? They are still sister chromatids. So that would be a good test question too, right? Which, and considering interphase, in what phase or phases, or which part or parts are the chromosomes singular? And that would be G1. Which, in what part are they duplicated? Like, or in which of the three are they double? Are they sister chromatids? And that would be S phase and G2. Anyway, at this point, since this is the last part of the interphase, the last part before it goes into the mitotic phase, which is all about duplication, this is really when it's starting to ramp up and preparing to divide. We got more organelles duplicating. The cytoskeleton, which I taught you about in chapter 3 and said it wasn't that important, but some stuff would come back later. It's come back later. right? So, that cytoskeleton's keeping things where they go, and it's keeping the cell in its particular shape. But since things are about to need to start moving around within the cell, and since the cell is going to need to change shape and eventually pinch into two different cells, obviously we need to get rid of that cytoskeleton. Because that's the thing keeping it the way it is, and we don't want it the way it is anymore, right? We're trying to change some things up. So any questions about the G2 phase? All right, you guys are nice and easy. If I'm going too fast, let me know. Don't be ashamed to say slow down or can you repeat that? That's good. I need that. Because I've been teaching this for years. So for me, this is like just easy and normal, but I understand for you, it's like, that's a lot of words put together that I've never seen before. So slow me down if you need to. All right. So this is where it gets a little bit confusing. I want to make sure people understand this. We're done talking about interphase, right? So we're done talking about that big interphase where it spins 90% of the time. We are now moving into the mitotic phase, and this is where I was saying there's a pre-lab question people get wrong a lot. So I'm going to go ahead and say this now. The mitotic phase itself is broken down into two parts. Just like the interphase is broken down into three parts, the mitotic phase is broken down into two parts. Now, with the mitotic phase, they happen to overlap instead of one after the other, like in interphase. But there's mitosis and there's cytokinesis. Those are two different parts of the mitotic phase, and people get that confused because mitotic phase versus mitosis, they sound very similar. But mitosis is just part of the mitotic phase, so is cytokinesis. So when you get a test question or a question on the pre-lab that says list the stages of mitosis in order, and I'm going to talk about that here in a second, do not say cytokinesis. Because yes, cytokinesis does come during the mitotic phase because they overlap. But cytokinesis is not part of mitosis. Make sure that's very clear. I'll slow that down. Cytokinesis is part of the mitotic phase, which is a two-part phase. But cytokinesis is not technically a part of mitosis. So when you're listing the stages of mitosis, you should not include cytokinesis. If the question is very specific and says that, what are the stages of mitosis? Now, if it says, what are the stages of the mitotic phase, then you would include cytokinesis. But we're talking about, it says specifically mitosis and not the mitotic phase, do not include cytokinesis. Anyway, after I was saying all that, what's the difference? What's the difference between mitosis and cytokinesis? So in mitosis, this is when the nucleus and its contents do divide into two, right? This is what we're after. We're after trying to divide this cell into two parts, right? That's what the whole mitotic phase is about, is taking one cell and turning it into two. So basically, if we're taking one cell and turning it into two, we're breaking that down into two parts. We're taking the nucleus and turning it into two, that's mitosis. And then there's taking the rest of the cell and turning it into two cells, and that's cytokinesis. So that's how that works. And generally speaking, again, like it says, they overlap. But generally speaking, mitosis starts the process, and then somewhere in your textbook it's specific, but I don't care. I'm not going to get that specific. I'm just going to say somewhere during mitosis, while mitosis is happening, cytokinesis starts doing its thing. So that would be two great, or at least one great test question, maybe two. Which of the mitotic phase, which of the two divides the nucleus in two? That's the mitosis. Which of the two divides the entire cell into two? That would be cytokinesis. Anyway, since we're talking about mitosis and not meiosis, the end product is two genetically identical daughter cells, and that will be definitely a test question. Because you're going to have to, some of these questions are about differentiating between mitosis and meiosis. So it'll say something like, which of the two between mitosis and meiosis produces two genetically identical daughter cells? And the answer is mitosis. Or more than likely, the question is going to say, mitosis produces what? And there'll be a number of options. And the correct one is going to be two genetically identical daughter cells. Some of the incorrect ones will be 4 genetically unique daughter cells, because that's the correct answer if the question was about meiosis, which you haven't learned yet. And then there'll be a bunch of nonsense questions like 4 genetically identical daughter cells, so nothing ever gets us that, or 2 genetically unique daughter cells. Like there's going to be some nonsense questions, but the only possible correct answers are 2 genetically identical daughter cells, which will be correct for this one, or 4 genetically unique daughter cells, which would be the one you can learn about later. So be ready for that. And sort of like in photosynthesis, this is a good reminder. When you get questions about photosynthesis, it's going to say, which of the 2 stages does this happen, right? And the only real answers are going to be light reactions or the Calvin cycle. Anything other than that is nonsense, and you'll get 0 credit. But for those questions, at least if you guess Calvin cycle or light reactions, you'll at least get partial credit. And most likely it'll be the same thing here. When I ask what are the results of mitosis, there's going to be 2 correct answers, 2 that were at least viable, and then the rest of them are junk. So as long as you get one of the 2 that at least makes sense, you'll get partial credit. At least that's my plan. I might have to go in and adjust that exam a little bit to make that happen. Anyway, that's the mitotic phase in general. We're going to talk about it more specific now. We're going to talk about mitosis followed by cytokinesis. So before we get into the details of that, does anybody have any questions about this slide? All right, so let's dig into mitosis and cytokinesis. Now, mitosis is broken down into 4 stages. I've got a little star there to remind me that your textbook is actually a little bit more complicated. Your textbook says that it breaks down into 5 phases. So for example, and I'm going to talk about this in more detail later. You can see here the first phase is called prophase. Your textbook says this prophase followed by prometaphase and then metaphase, right? So your textbook has a whole extra thing in here. But I'm keeping things simple because this is a 100 level class and it's not meant for biology majors, so I want to keep it more simple. So we're looking at these 4 distinct phases. Forget about the one between prophase and metaphase. So there they are, and there they are in order. There'll be 2 different test questions that people mess up. The first one's easy, and it's going to say, what are the 4 stages of mitosis in the correct order, right? So you're going to have like at least 4 options with these 4 words in different orders. And the correct answer is prophase, metaphase, anaphase, telophase. That is the correct order. And then I'm going to talk about this later once I've taught you all this stuff. But then there's going to be one that says, in this phase, the chromosomes do this in blank phase. Then the chromosomes do this in blank phase. Chromosomes do that in blank phase. That'll be like a fill in the blank, and it will not be in order. And that's usually the question they miss, because one of the options for that question is going to be prophase, metaphase, anaphase, telophase, and people are going to guess that because it's the correct order. But that question isn't asking about the correct order. It's saying, in this phase, this happens, in this phase, that happens, so on and so forth. So keep that in mind as I'm teaching this, and don't let that test question fool you. It's not a trick question. It's not meant to be anyway. Anyway, again, each of these phases is about the movement and separation of chromosomes. Some other stuff happens in that as well, but mostly that's what we're going to focus on. That's what's happening with the chromosomes. For extra credit, you can look up other stuff too. So I'm going to tell you what happens. Well, prophase is a bad example. Metaphase, right? That's almost the entire discussion is going to be about what the chromosomes did. So for extra credit, you can look it up and tell me what other things are happening in the cell while this is happening. And we know what the chromosomes are doing, but what else is happening? Anyway, we're going to talk about them separately. Again, you can study how you want, but I recommend starting with the big picture and then adding details. In this case, that would mean memorizing that these are the 4 phases in that order of mitosis, and then starting to add the details of each one. So, like I said previously, or said right here, we're talking about the movement of chromosomes, right? That's mostly what we're concerned about is the movement of chromosomes. What are the chromosomes doing in these different phases? And like I said earlier, slides back when we were talking about interphase, I said that the centromeres had been created, right? They had doubled up. So now you have these 2 centromeres, and I said those 2 centromeres create something called the mitotic spindle. So the mitotic spindle is the part that's actually going to move stuff around. This is a football-shaped structure of microtubules. Remember, we learned very quickly, we went over microtubules in chapter 3 And I said, you're not really gonna get any choice questions on this for exam 1, exam 2, but later it's gonna come back, so there you go, it's come back. Not that you need to know all the details that I taught you the first time about microtubules, you just need to know it's part of the side of the skeleton, it's what we're using to move things around. Specifically in this case, even though they move other things around, we're only concerned about them moving around the chromosomes. So again, when we start talking about prophase, metaphase, anaphase, telophase, the question is going to be, the focus is going to be on what the chromosomes are doing in each of those stages. And just so you know, because I don't know, I'm probably not going to remind you each time, but every time, it's these mitotic spindle that's helping them move around. Any questions about that? Okay. I'm probably not going to specifically have a test question about the mitotic spindle, but I'm going to use that term a lot in test questions and in this lecture. So that's why it's important that you know what it is. Another good extra credit thing you could look up is, how long does it take to form the mitotic spindle? Does it depend on the cell type? Does it depend on the species? Because remember I showed you that one slide that had all those different chromosome number of species. That rat had like 100 something. The deer, I forgot how many it had, maybe like 4 or 6. So obviously the more chromosomes you have, the more mitotic spindle you need. So you can look that up for extra credit if you want. So anyway, yeah, that's the mitotic spindle. Now let's talk about prophase. So remember, this whole thing is about separating chromosomes, right? And let me ask you this, what do we have here? 9, 3, 6, 9. Here we have, it would be better if it was 10, but whatever, let's pretend like that was an even number. Here we have 9 balls of yarn. Here we have the same 9 balls of yarn. Now let's say I gave Champion the one on the bottom right. There's 9 balls of yarn. I gave Stover this Ravelly 9 balls of yarn and gave it to her right there. And I said, all right, I want you guys to race. I want you to put half of your balls on one side of the table and half of yours on the other side of the table. And I want you to do the same thing. Who do you think is going to win that race? The one with this yarn or the one with this yarn? Who's going to win the race of getting their yarn separated half and half? Yeah, Champion, right? And why is that? Yeah, it's not a big tangly mess, right? So if you remember what I talked about Wednesday, maybe Monday, remember I said your chromosomes prior to S phase, or prior to the mitotic phase, sorry, big squiggly mess, right? They're not condensed. Remember we talked about chromosomes when we talked about eukaryotic chromosomes? I said eventually they get condensed and you can see them under the microscope. That's the situation, right? This is what your chromosomes look like before mitosis. Now, obviously, if we're about to split them up, then your cell is like, all right, well, we need to get organized. So in prophase, they turn them into this. I'm hoping that, again, like I said, I hope this is very intuitive for a lot of you. And that should be intuitive. It should be intuitive that in the first part of this process, we need to get our chromosomes ready to move because that's the whole idea of this process is to move the chromosomes. So it makes sense for the first part of it is to get those chromosomes ready to move. So let's talk about, this is what happens in prophase, and you need to know it. So the first bullet point we just talked about, the chromosomes get coiled up. And again, I'm hoping that's very intuitive. That should make sense. We want to get them separated. Well, we need to get them ready to move. The second bullet point is not really something that's necessarily new to this. It's just a reminder of where we're at right now. Each chromosome exists as sister chromatids still, because remember that happened in the S phase, so they're still together. So that's not necessarily me telling you, hey, this happened during prophase. It's just a reminder that that's the current situation that we're in, that we have been since S phase. Here's another thing, and I know you're taking notes. Let me back it up again. Again, if I was doing this race, but this time let me change it up a little bit. I got a Kroger bag full of these yarns, and I've tied it up, and I give it to Champion. And I've got a Kroger bag full of this. I've tied it up and I've given it to Stover. What's the first thing they're going to do when I say I need you to separate those from different sides of the table? I take it out of the bag, right? Intuitive. Well, the bag is your nucleus. So obviously, if you're going to separate these chromosomes to different sides of the cell, you need to get rid of the bag that's holding them. So that's this bullet point. The nuclear envelope, as far as we're concerned, disappears. Your book explains how it disappears. You should learn it maybe for extra credit. because I might ask you some questions about that. But as far as we're concerned, for the exam, in your brain, you can just think, all right, the nuclear envelope has just disappeared. So obviously not true, right? It has to go somewhere. Something has to happen in all those pieces, but to keep things simple for this 100 level class, you can just think in prophase, the nuclear envelope disappears. And again, I'm hoping this is very intuitive, because if we're going to separate things, you got to get rid of the thing that's keeping them together. Speaking of which, if we're going to separate things, you've got to attach them to the thing that's going to move them around. So that's the other thing that's happening in prophase. The spindle, that mitotic spindle that I talked about, it's finally attaching to the chromosomes. Specifically, and I'm not going to get this specific on the exam, but remember, when I talked about those chromosomes, trying to draw it, we had those sister chromatids. Actually, no, there's one already drawn right here on the right, bottom right. So there you've got sister chromatids on the bottom right, right? And I said that sister chromatids are attached in the middle. It's something called the centromere, and I told you that word's going to come back again. So there it is, it's coming back. So yeah, that's where the centromere is. That's where they're attached. So the mitotic spindle is attaching right there, right? From one side here, one side over there. The mitotic spindle is attaching. Again, I'm hoping this is intuitive, because this would all make sense. If you're going to move chromosomes around in this whole process, then the first step should be attaching the thing that's going to move them around. Now, finally, this is where this is a little bit new information. It's a little less intuitive. The chromosomes at this point, after they've been attached to the centromere where they can move around, they start moving towards the center of the cell. So that at this point, they're moving towards the center of the cell. They haven't gotten there yet, but they're moving towards the center of the cell. So that's it for prophase, the first part of this process. Does anybody have any questions about prophase? All right. Oh yeah, I've got a little note for myself for those two stars. Technically, the spindle is attaching to the chromosomes and the centromeres in prometaphase. In your book, it even goes as far as to mention there's a protein complex called the kinetochore, excuse me, but you don't need to know that. So again, if you're taking notes, you write that down. Like, you need to know this easy version, not what the book is teaching you. You don't even know, I'm probably not even going to ask you where the spindle attaches to the chromosomes. It's just that when the test question comes, I might say the 10 spindles have attached to the centromeres, or I may say that they've attached to the chromosomes. Or I may say that they've attached to the sister chromatids. Either way, I'm saying the same thing in different ways. All right, so that's prophase. Let's see if anybody can guess what defines the next phase. And I'll leave it, or give you a hint. Last thing I'm telling you about prophase is that at the end of prophase, the chromosomes are starting to move towards the center of the cell. So what do you think defines the next phase? Forget what it's even called. But if the chromosomes are moving towards the center of the cell at the end of this phase, what do you think is the defining thing for the next phase as far as the chromosomes are concerned? And hopefully this is common sense. Don't overthink it. This is the end of this phase. So what do you think is happening in the next phase? What defines it? What is it that the chromosomes do? You think that they're like, oh, that must be the next phase. And we're officially calling this the next phase. Don't overthink it. Let's look at that last bullet point.
Is it where they line up?
Yeah, perfect. Or, and that is, yeah, it's a great way of saying it. And that is one of the ways I'm going to say it. In the context of this, yes. So if here at the end of prophase, they're moving towards the center of the cell, then once they get to the center of the cell, that is metaphase. Don't forget that picture. No, I'll get there in a second. But yeah, I'll back up to this. So yeah, once they get there, like she said, once they're lined up, that is metaphase. But we'll come back to that really quickly. Let's look at these pictures of metaphase or prophase. Actually, first we can look at interphase. These pictures are from the old textbook, not yours. This is regular old cell, right? We haven't gone through prophase yet. We're just doing our regular interphase thing. Like I said, the centrosomes have been, they've doubled. They're ready to go. What you can't see is they're starting to move this way, right? The chromosomes haven't coiled up yet. The nuclear envelope hasn't disappeared. Everything's just business as usual. Then we get to prophase. The mitotic spindle is still forming, right? These things are still moving towards the opposite ends of the cell, which you can't see because it's not animated. The chromosomes have coiled up, so we have like visible chromosomes now. You can see where the centromeres are at the middle of them. Eventually, And technically this is prometaphase, but again, we're not going to get into that. We're still just going to call this prophase. Eventually, these mitotic spindles have attached to the centromeres, right? They've attached to the sister chromatids, and they start to move towards the center of the cell, right? If this was animated, that's what you can see it happening, right? All those things starting to move towards the center of the cell. And like I said, if that's the end of prophase, when they're starting to move towards the center of the cell, then as Stover correctly said, in the next phase, that's kind of how we define it. Once they get there, then we call that metaphase. And the book, describes it like this. The centromeres are lined up between two poles of the spindle at the metaphase plate. Nice and easy though, for the exam, I'm just gonna say they made it to the middle. They're lined up in the middle, something simple like that. And then the easy way to remember that, I think, would help me as a student is that I remember metaphase. Of course, meta means something different than what I'm about to say. But I just focused on the met of meta. So I always tell myself, all right, this is the part where all the chromosomes met in the middle. So prophase, when I think of pro, I should have said this earlier, when I think of a pro, I think of like a professional athlete or something like that. Like they're number one. So I always remember that prophase was the first part. And then I remember metaphase is when they all met in the middle. So there we go. And there's a picture of it, right? You can see they've all met in the middle. What you can't see in this picture is now that they're in the middle, now these mitotic spindles are kind of pulling at them. It's a tug of war, trying to split those two sister chromatids up. So at the end of metaphase, that's where we're at. They're lined up in the middle, but these chromosomes are pulling on them and trying to separate them. So just like last time, let's see if somebody can guess what the next one is. But this is the end of metaphase, when these mitotic spindles are trying to separate these sister chromatids. How do you think we define the next phase? What's going on in the next phase? What happens to the chromosomes? We're like, oh, yep, that's now the next phase. What do you think? Again, focus on this last bullet point and don't overthink it. Well, no, focus on this middle one. Spindles are pulling those, they're pulling it. It's a tug of war. They're trying to separate them, but they're still together. So what do you think is the next phase? Oh man, you got it the first time. So in prophase, they were moving towards the center of the cell. Right, that was prophase, and then once I got there, that's metaphase. So now I'm telling you, in metaphase, the mitotic spindle's trying to separate them. So what do you think the next one must be? Don't overthink it.
They're pulling them apart.
Yes, well, this is a semantics thing. I think you're right. So here they're pulling.
Or,
Yeah.
separating.
Yes. And so the next part they've done their job, right? So the next part they've actually separated. I'm hoping again this is very intuitive to you. At the last phase, they were pulling at them. It was a tug of war. Well, once they separated, that is now considered anaphase. The sister chromatids are now separate. Therefore, they're no longer called sister chromatids. Technically, they're called chromosomes, right? They're now separate. And actually, before I even get to the next bullet points, I won't give that information yet. Okay. So yeah, that's nice and easy, right? Anaphase. Anaphase is when they finally got separated. So here's a question you can look up for independent work. Do we consider anaphase once the first chromosome is separated? Or is it considered anaphase once they've all separated? Or does it all happen at the same exact moment? Or is there something in between? It's like, well, 50% of them have been separated, so we're calling that anaphase now. You can look that up for extra credit if you want. Anyway, yes, anaphase is defined by the fact that they're now separated. So for the test, that'll be the question. Like, in which of the four phases are the chromosomes separated and turned in, or which of the four phases are the sister chromatids separated and considered full-fledged chromosomes? And that would be anaphase. So here we go again. Let's see if we do the guessing game. Towards the end of anaphase, because they've already been separated, now they're moving towards the opposite ends of the cell. Actually, before I get to that, I should really swap these. While all this is happening, the poles of the cell, so the cell started off circular or square if it's a plant cell. Anyway, let's just call it circle to keep things simple. While all this is happening, the cell's also becoming elongated. It's turning more into an oval than a circle. That's what this last bullet point is. But let's focus on this middle bullet point before we move on to the last phase. I'm telling you here, the end of anaphase, that the chromosomes are moving towards opposite ends of the cell. Opposite poles of the cell, however you want to say it. So what do you think happens in the last phase? If the chromosomes at the end of this are moving towards the opposite ends of the cell, what do you think the next phase is? There's a pattern here. At the end of prophase, they were moving towards the center of the cell. Once they got there, we called it metaphase. Then in metaphase, there was a tug of war to separate them. Once they separated them, we called that anaphase. See the pattern? Now, in anaphase, they're moving towards the opposite ends of the cell. So what must the next phase be?
Something bloom after.
They do, then we'll get there. But just focus on this bullet point right here. This is what's happening at the end of anaphase. So what do you think is happening at telophase? At the end of anaphase, they're moving towards opposite ends of the pole, opposite ends of the cell. What must the next phase be? Yeah, but basically they're there. They make it, right? So at the end of anaphase, they're moving towards opposite ends of the pole. So guess what? Once they get there, that's called telophase. There we go. Telophase. At this point, we now have two groups of chromosomes, right? They've been completely separated. They've reached opposite ends of the cell. And then the easy part to remember, or the easy way to remember the parts of telophase is this is the opposite of prophase. Because now the deed is done as far as the nucleus is concerned, right? We've separated the chromosomes, sister chromatids, excuse me. We put them back in their spots. So now we can just undo everything we did. Now we can put them back in their bag, so to speak. Right now, the nuclear envelope can form back up because we're done separating them. So we need that nuclear envelope to keep them safe, like they usually are. The chromosomes no longer need to move around, so they can uncoil. Right? They don't need to be coiled up. They need to be uncoiled, and that'll make more sense in a later chapter when we talk about how we read the DNA code and do things with that. But we'll talk about that later. We're done moving them around. So the stuff that we use to move them around, we don't need them anymore. So the spindle disappears, right? We don't need that anymore. And mitosis is now considered complete, right? Sister chromatids have been separated. We now have two new nuclei. Mitosis itself is complete. Keep in mind, however, that we just got finished talking about mitosis. And mitosis is one of two parts of the mitotic phase. Mitosis is the part that takes care of the nucleus, which is done. We haven't even mentioned much about cytokinesis, which is the part that splits the cell into, right? So that's probably where we'll end it. Let me think. Yeah, so when we come back, we'll talk about cytokinesis. The last word for attendance, say TL, another musical birthday, it's Tommy Lee. Yeah, so there you go. Extra credit if you when you send those words in, if you tell me which band Tommy Lee played for and which band Lindsey Buckingham played for. And that's it. We're done 1 minute early. Woo, enjoy your weekend. And don't forget to have office hours until 12. If you need help with the P-Lab or if you need help with the lab, please let me help you. I prefer it. You don't copy off somebody who doesn't know what they're talking about. Instead, you can get the correct answers from me. And don't forget, if you're going to send those words in, as always, you got to send them in before 9 a.m. Oh, thanks. Right, can you send me an email line? I know you just told me that. That way I can have it in writing and I can reply to you.
And it probably happened to everyone.
Yeah, well, no, some people got full credit, which is why I need to look into it.
Okay.
So if I remember correctly, and I might not, so I'm gonna look into it, those have 0 points attached to them. The question about your name has 0 credit points, but I'll look into it just in case. Yes, ma'am. Thank you.
I don't know if it's just for me where we don't do it
That might be, yeah. Do you mind sending me an email? I know you just told me.
Yeah.
But it's, I have to have that, my email's like my to-do list. So, yeah, if you send me that, I'll take a look at it and fix it and let you know when it's fixed. Thank you so much.
Thank you.
And if it happens to be that that's like that for everybody, then I'll also give you a little extra credit for bringing up that, that glitch.
Okay, thank you.
Thank you.